welcome to my channel Sam Biology Class. Friends, today we will discuss about reverse genetics. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Reverse genetics has emerged very recently in the beginning of the 1980s. Since the late 1990s, tens of eukaryotic genomes have been sequenced. Genome-wide sequencing revealed a large number of genes whose functions are not known and cannot be predicted. Assigning function to the genes is important. Reverse genetics find out the function of genes whose sequences are known. High-throughput reverse genetic approaches therefore play a major role in post-genomic era. Reverse genetics is a scientific method that investigates the function of a gene of known sequence by creating a specific change in that gene to see what effect it has on the organism's phenotype. Phenotype is the observable characteristics of the organism. Genes control specific phenotype through the process of gene expression. So when the function of a gene is altered or the gene is silenced, the phenotype is also changed. By analyzing the resulting change in the phenotype, the function of the gene can be determined. In reverse genetics, a gene of known sequence is the starting point. In this approach, researchers first identify the gene of interest, then modify its sequence, and after that, they study the phenotypic change. By analyzing this phenotypic change, the function of the gene can be determined. By contrast, forward genetics, also called classical genetics, starts from a known phenotype and then works backward to discover the gene responsible for that phenotype. Reverse genetics therefore proceeds from the genotype to the phenotype. Forward genetics, on the other hand, proceeds from a known phenotype to the genotype. Now the question is, why the name reverse genetics? This is because its direction is opposite to that of forward genetics. The concept of reverse genetics was first formulated by Charles Weismann in 1978. Weismann was the first practitioner of reverse genetics implementing site-directed mutagenesis (SDM). Reverse genetics starts with the identification of a gene whose sequence is known. This is the first step. In second step, the sequence of the gene is intentionally modified by different techniques such as gene knockout, gene knockdown, site-directed mutagenesis or CRISPR-Cas9. These are the tools of reverse genetics. When the sequence of a gene is intentionally altered, the phenotype is also changed. By analyzing the resulting change in the phenotype, the function of the particular gene can be predicted. For example, one gene of a cell was knocked out. After gene knockout, it was found that cell division has been disrupted. This indicates that the particular gene is involved in the process of cell division. Another example, one gene of a virus was silenced, after which the virus became less pathogenic. Based on this observation, we can say that that particular gene contributes to the pathogenicity of the virus. Because of this, when the gene was silenced, the virus became less pathogenic. The same principle is applied in the production of attenuated virus vaccine. Tools of reverse genetics. First one is gene knockout. Gene knockout is a technique used to make a gene non-functional to study its function by observing the effect of its absence. This is often achieved by introducing a specific mutation that prevents the gene from being transcribed or translated correctly using methods such as CRISPR-Cas9, TALENs or homologous recombination. Second one is gene knockdown. It is the process of reducing the expression of a gene. Genes can be knocked down by RNA interference. RNA interference is a method that uses small interfering RNA to guide a cellular complex to degrade specific mRNA, preventing its translation and reducing the expression of the particular gene. Third one is site-directed mutagenesis (SDM), which is a technique used to introduce a specific intentional change such as insertion, deletion or substitution in the sequence of a gene. Another one is CRISPR-Cas9. It is a gene editing technology that uses a combination of guide RNA and Cas9 enzyme to introduce a precise cut in the DNA. This process allows for genome editing. Reverse genetics has several applications such as understanding disease and developing vaccines. It is also applied in agriculture to modify plants or animal genes to improve traits such as yield or disease resistance. Another application is the investigation of the fundamental roles of genes in various biological processes. Friends, this is all about reverse genetics. For more updates, subscribe my YouTube channel, Sam Biology Class.